Okay, so guys, we're going to look at the internal structures of the leaf. We can see that we have the upper and lower epidermis, mesophyll tissues, and then we have the conducting tissue. So we have upper and lower epidermis, the mesophyll tissue in between, and then this conducting tissue, and the conducting tissue contains xylem and phloem to conduct the water and the phloem. Water into the leaf, phloem is taking food away to be stored in the roots and the stem. Okay? Everyone happy so far? So let's have a look at the epidermis now. The upper and lower epidermis of the leaf are covered by our epidermal tissue. And remember, our epidermal tissue has got guard cells. But the dorsal surface will not have guard cells. The guard cells will be only on the ventral surface. Okay, so dorsal surface won't have, ventral surface will have. These cells appear very brick-shaped. So what we have, grade 10s, is we have a layer of epidermal cells tightly packed on each other. There is a nucleus present. Now, if I could, unfortunately, it's quite unclear to see all of the cells. We can only see our guard cells in our microscope work, but it's fine. At least we got to see something. Yes? Okay, so we have our epidermal cells that are tightly packed, one on each other, next to each other. No intercellular air spaces. The nucleus doesn't always have to be on one side of the cell, by the way. It can just be random. I seem to have drawn it all on one side, so let's draw it there for fun. Okay, are we all happy with the epidermal layer? So this is the top epidermis. The epidermis protects the underlying tissue. So there's no intercellular air spaces, and it protects the underlying tissues. The regular epidermal cells do not contain chloroplasts. So no photosynthesis is happening in this particular layer. In actual fact, this layer is transparent. Okay? So it is a transparent layer. And the reason why it is a transparent layer is because it needs to allow light to penetrate to the lower tissues of the leaf. Okay? And so we have, because what is the function of the leaf, guys? Remember, what are we trying to do? We're trying to photosynthesize, right? And what do we need for photosynthesis to happen efficiently? We need light, okay, sunlight. And that is going to then penetrate through these cells, because it is transparent, to get to this lower layer of cells that actually does the work. Okay, so there's our epidermis. Over and above our first layer of our epidermis, we also have a cuticle. And I said to you guys, if you feel a leaf, the leaf does feel waxy. Okay, so ideally I'd like to pass this around, but then we're going to sanitize between, and it's just not going to be worth it, okay? But anyway, we have a cuticle that sits over this layer of cells. Oh, I drew it a bit fat there, but anyway. Is everyone cool with that? I just want to move this wire because I can see me getting tripped up on it. Which might be fun for you, but not so much fun for me. Man, we would never laugh at you. We would be concerned if you tripped and fell, man. Right, the waxy transparent cuticle covers the epidermis and it reduces excessive water loss while still allowing light to penetrate. So we have this cuticle. What does it do? It prevents transpiration. We know that transpiration is the loss of water by plants. Okay. Any questions there with the upper epidermis? It's really simple. Right, let's go lower epidermis. So the lower epidermis obviously has guard cells. So the epidermal tissue is made up of two types of cells, epidermal cells and specialized or modified epidermal cells called guard cells. And that's what we got to see under the microscope. Okay? Guard cells, which form an opening between two guard cells, is called a stoma, S-T-O-M-A, single, stoma, plural, stomata. And these are only going to be found on our ventral surface. So let's draw our epidermal layer. So we have epidermal cells. We would have guard cells kind of found between. And these guard cells are surrounded by epidermal cells, by the way. Now I can't do it nicely. Sorry. Made a mistake. Yes? Yes. Please sanitize. When you go now, sanitize. And then when you come back, sanitize again. Okay, so we have, I'm not going to draw the epidermal cells around the guard cells. Are you happy, grade 10s? Yes? And then we'll have epidermal cells. Let's have another one. And then we would have a guard cell. Again. 2 guard cells next to each other. And this is what we saw. Yes? And what do the guard cells allow for? For gaseous exchange. Okay, so that's stomatal pore. So these are the individual guard cells. Guard cells. And that pore is the stoma. Okay, the stoma. And the stoma is what? Many stomata. Is everybody happy? That is where gaseous exchange is going to take place. How do you spell exchange? Exchange. So much laughter. And my brain works faster than yours, that's all. Gaseous exchange, are we all happy? Now, the thing about these guard cells is that they do actually contain chloroplasts. Kind of odd that the epidermal cells do not have chloroplasts and there's no photosynthesis happening there, but these guard cells do have chloroplasts. And so I'm going to just put them in. Yes? Now what the guard cell does is it will close the stomata on a hot day to decrease water loss and open the stomata when the temperature is not as excessive and when it can regulate, okay? So it's responsible for regulating. So inside of your guard cell, we have a vacuole. Plant cells have got vacuoles, yes? So I'm drawing it in blue. The vacuole, okay? When we have water loss, so in other words, when there is a high amount of transpiration, when would we have a high amount of transpiration, grade 10s? When it's hot, right? Okay? So midday, 12 o'clock, really hot, we've had excessive water loss. What will happen is because these inner walls, and if you actually look at the picture that I took, oh, now my phone's on it. Okay. Anyway, if you look at the picture, you'll see the inner walls are a little bit darker. Did you see that it was darker? That's because the inner walls are thicker. And so what happens when we lose water, these thick walls start to pull towards each other. And they will close the stomata to decrease loss of water, to decrease transpiration. So the guard cell is responsible for regulating water loss. In other words, regulating transpiration. Okay. It will close the stomata to prevent water loss, even at the expense of gas exchange. It's kind of like it's got a balance. Cool, I need gas exchange to happen, but I can't have it happening at the expense of water, because we need water for photosynthesis. So it's regulating this quite carefully. Sponga, are you awake, my baby girl?
Yes, I'm
You sure? You look a little bit zoned.
No.
You sure? Okay. Do you all understand this concept? Remember when we did guard cells, we said that the inner walls are thicker than the outer walls, and when it loses water, it'll start to collapse on itself. We spoke about that concept of the vacuole holding it open and turgid. But now, because the vacuole's lost water, it starts to collapse and shrink. Everyone get that? Yeah? We spoke about that concept. And it will then close that, decreasing water loss. Happy days? Right, so the lower epidermis cells contain stomata. Guard cells are responsible for opening and closing the stomatal pore. The guard cells are bean-shaped, done it, and in surface view, and have thick inner cell walls. So the inner walls are thicker than the outer walls, and what that does is it'll close. There is chloroplast, so they do have chloroplast, so they are able to photosynthesize, but our epidermal layer does not. And by the way, there is also a cuticle on the lower epidermis, just so you know. So there will be a cuticle here. Yeah? Cuticle, upper epidermis, lower epidermis. Gaseous exchange, guard cells, boom, boom, boom. Yeah? The stomata are responsible for gas exchange during photosynthesis, respiration, and transpiration. And so there you can see that's just an illustration of it with the epidermal cells all the way around. This is what we saw. Okay. I think we covered guard cells. I don't think we need to go through all these pictures. Are we happy with guard cells? Are we happy with epidermal layers? So upper epidermis is called the dorsal epidermis. This is called the ventral. Guard cells are on the ventral surface because it's shading it from direct exposure to sunlight. Are you sleeping, Kamuelo? What's wrong? Your face. What's it doing? You don't know what your face is doing? What's happening to your face, Sunny boy? It feels like you just put, maybe you have a waxy cuticle. Right. The tissue found between the upper and lower epidermis is called the mesophyll tissue. Okay, so the mesophyll tissue makes up the majority of the bulk of the leaf. Okay, I know that when we look at a leaf in this view, we kind of think, what bulk? There's nothing there. Agreed? Okay, but microscopically, there is evidence of guard cells and there is evidence of tissues. Okay, and so we have between the upper and lower epidermis, we've got a whole bunch of tissues. I want you to quickly look here. Can you see a difference between this layer of cells and all of these cells?
Yes.
Can you tell the difference?
Yes.
Yes. What is the difference between this layer of cells and this layer of cells? These ones are very tightly packed together, 100%, while these ones are, there's air spaces between. Okay, that's the one difference. The second difference is the shape. Do you see how these are kind of irregular, rectangular shape?
Yeah.
Okay, so this is called the mesophyll tissue. We have two types of mesophyll tissue, what we call palisade mesophyll and spongy mesophyll. What do you think, which one do you think is going to be called the palisade mesophyll? The top ones that are like this, right? And these ones are the spongy mesophyll. Is it easy to identify?
Yes.
There's a clear difference, right? So when let's talk about this upper mesophyll layer. The upper mesophyll layer fits right underneath your upper epidermis. Okay? It's made up of cells called palisade mesophyll cells. They are really kind of irregular shape, looking like rectangles. Okay? They have got lots and lots and lots of, what do you think they're going to have lots and lots and lots of? Chloroplasts, yes? Yes? So all of those, these little dots are actually chloroplasts. So I'm going to draw them the same way. Okay, obviously we know the structure of a chloroplast. Yes? Do we?
Yes.
Some of us do. Not dissing you. I love you. Okay, chloroplasts, lots of chloroplasts. Are we all happy? These cells are tightly packed to each other, next to each other, and that's going to give them greater exposure to light. So they're very, very tightly packed, lots of chloroplasts, lots of exposure to light, lots of photosynthesis happening. This is the business end of this whole process. So that is called your palisade mesophyll. Grade 10. Have any of you heard of a palisade fence?
Yes.
You heard of a palisade fence? What does a palisade fence look like? It's got all of those. There's the palisade fence, so when you leave, you can look outside the window. Okay? If you don't know what a palisade fence is, it's got the fence parts all next to each other like in rows like that. That's what this is kind of resembling, except for the fact that there's no space between, they're just one next to each other. Tomorrow, grade 10, we will do spongy mesophyll. Oh wait, 2 seconds. Spongy mesophyll is made of cells that have got lots of air spaces. Do you agree with me?
Yes.
That's why it's called spongy mesophyll. 2 seconds. What is the purpose of those air spaces? What happens at the stomata, guys? Gas exchange. Where's the gas needed? Up here, so it needs to go through this to get to there. So it goes through. Isn't that exciting? Isn't that clever? They're these little air spaces that allow for CO2 to move, because here's where we have gas exchange. It's moving through the spongy mesophyll to get to the business part. Where is this? What process is happening here? Photosynthesis.
I like this.
Okay. Thank you for those of you that are working at home. You need to work. Goodbye.